That's where I started working with cryopreservation, which I'm going to be covering today. Some of the species that I worked was southern flounder, channel catfish, and probably the most difficult one was the green swordtail. Why? Because I was freezing the sperm and doing artificial insemination with the frozen sperm to these guys. They are really little. So I successfully produced some offsprings using cryopreserved sperm. And also I work with other marine species, such as red drum, red snapper, and the spotted sea trout. Then I did my postdoc in Kentucky State University, so I went to Kentucky. Now I work with this fish. Are you guys familiar with this fish? What is it? A paddlefish. What is so unique about this fish? Yeah, that's also unique. Yeah, it's a filter feeder. And what they do over there is what is important, like the female, what the female can produce. It can produce caviar, just the same as an sturgeon. So basically what they do is something called reservoir ranching. So are you guys familiar with ocean ranching? So this is done in the reservoirs. So you stock the paddlefish, wait for 8 years, because it's going to take 8 years for the female to mature, harvest those, and get your caviar. You know, that's the money. You can make a lot of money with the caviar. So basically now what you want is to have females. So one of the techniques that I was involved is called direct gynogenesis, which is using the sperm from a donor species, in this case an sturgeon, UV radiated, so you can remove the DNA and just use it to activate the egg of a paddlefish. And you want to retain that polar body of the egg by shocking the eggs at high temperatures. By doing this, you can increase the percentage, like 80% of the females. And also, if you are able to identify this one, then again, you can produce all females. And well, this is different as the humans, so the heterozygote is just like the birds, it's a WC. So another project that I was involved was reusing decommissioned facilities. Nowadays, there is a lot of new wastewater treatment plants, and the old ones, they demolish. You know, they spend a lot of money building all those, and at the end, they just decide to get rid of those. Why? They hold water. You know, they have ponds, they have tanks, and you can easily convert it for fish farming. So what we did is use some of the decommissioned facilities and convert it to aquaculture products. So this is the hybrid striped bass. It's a cross between the striped bass and a white bass. And they do this because the hybrid bigger. It grows faster, it can live in fresh water, and it tastes pretty good. So that's why the industry, a lot of the aquaculture industry, is working towards these hybrids. And like I say, if you look around, there is going to be a lot of opportunities for you in reusing some of these tanks. One of the advantages in specific for this project was that we were working in a wastewater treatment plant, and we were using their treated effluent. Nowadays, EPA has really strict regulation on the water. So it's really clean, that water. So why don't we use, instead of dumping it into the river, why don't we use that water for aquaculture? So we were raising the fish in the treated effluent that is also disinfected, and we evaluate for the contaminants on the fish, and everything is safe. So they are going to be safe to eat. So you may wonder, why did I ended up at Humboldt State University? Well, because in Arcata, during the 70s, there was this well, there is this wastewater treatment plant, but there was some ponds that they were doing what I was doing during my postdoc, which was using the effluent from the wastewater for aquaculture. So George Allen, he was the one during the 60s that decided to do this. So I came to Humboldt State trying to continue this project. Unfortunately, the city of Arcata say, you know, we don't really want to continue doing that. They say that they need the space to have more oxidation ponds for the wastewater treatment plant. So unfortunately, I ended up without, you know, the project that I was thinking that I was going to be doing. So that's why I came here. I'm looking for collaborations. I'm looking for ways that we can work together. And I'm just going to show you some work that I did for my PhD on cryopreservation. And I have the title Ex-situ. So let's remember conservation, in situ and ex-situ. So basically, on in situ, we want to protect that environment, that natural habitat. In ex-situ, we are removing either the individuals, the embryos, from outside their environment. And we can find a lot of examples on in situ conservation. You know, we can have habitat restoration, the protected areas, but none of these we can rely that we can save that species. We need to start looking in something else. And that's where ex-situ come into play. So ex-situ is also divided into two parts, in vivo and in vitro. In vivo is going to be those individuals that are going to be staying in aquariums, or in vitro is cryopreserving the sperm or cells for having a germ bank. And I'm going to be talking more about that. Some examples, Monterey Aquarium, definitely, there is a good example of ex-situ in vivo. So what about the in vitro or frozen zoos? Where we can find some of those? And why do we want to do ex-situ? Well, again, we want to have those genes. We want to bank them. We want to have the backup. And the most important part, there has to be complementary to the in situ approach. So they have to work together. Now, well, I already talked a lot about what is cryopreservation. Well, it's pretty simple, you know. We just want to use very low temperatures to preserve the integrity of the cells. How do we do that? By using liquid nitrogen. It's pretty cold. You know, it's get minus 196 degrees Celsius. At this temperature, you can preserve for hundreds of years those samples. How can we protect a cell or a sperm cell from damaging during the cooling process? Because, you know, during cooling, the ice crystals are going to be start forming, and that's going to cause some damage to your cells. So the same as your car, you use antifreeze. So we use antifreeze, propylene glycol, you can use methanol, alcohol base, and everything is going to depend on how toxic they are to the cells. You add those. So now the environment is hypertonic, so the cell is trying to osmoregulate, and some of these cryoprotectants ended up, depending on your concentration and the time of exposure, ended up inside the cell. Now, you start cooling down, which is another factor. How fast do you want to cool down those cells? Ice crystals start forming. The same is going to be a hypertonic environment for the cells. And lucky, you can reach this glass formation temperature where there is no more ice crystal forming. And in this way, you can preserve the cell or the sperm cells for centuries. let's put this into perspective. Why do you want to preserve the sperm? Well, just think about it. You have a farm. You have a male broodstock of an sturgeon that probably is 15 years old or older. So it's a really valuable to you. So one time you forget, you remove the standpipe, and you forget to put it back in. Next day you come, and that's your fish, you know, your favorite fish. You love this fish, was your best breeder. Now it's gone. You try your best to resuscitate, even probably mouth to mouth, I don't know, but it's gone. But if you were thinking ahead and paying attention, and you were cryopreserving some of his sperm, then it doesn't matter. You can eat it for dinner and use that sperm forever. So why I say sperm and not embryos? Well, the fish embryo, they are relatively large size compared to a sperm cell. Also, they have a complexity, a multi-layers, that is difficult for those cryoprotectants to go and penetrate all those layers. So unfortunately, there has not been successful in cryopreserving fish embryos. Just to see how important is cryopreservation, USDA has the National Animal Germplasm Program. And this program is meant to have a gene bank, a frozen bank of either blood, semen, or embryos. So let's see how many freshwater fish are out there. Just 33 species they have in that bank. What about marine fish? Just 6 species. You know, they don't have nothing on marine fish. What about invertebrates? Just 5 species, 3 of those being coral. So as you can see, it seems that, where is the abalone? Why don't we have a backup over there? Where is the mixed method? Nobody knows about it? Or we have to start backing up a lot of the germplasm around here. And if you compare it with cattle, with 2 species, with almost 500,000 of samples. So overall, here in this bank, aquatic vertebrates and aquatic marines, they are not even represented. I'm going to have this talk, I'm going to divide it with 3 examples. So the first example that I'm going to giving you is going to be the catfish, and how cryopreserve help the catfish industry. Well, the first thing that you need to know is the catfish is the #1 aquaculture product in the United States. Unfortunately, in the last few years, we have a decrease in the production. So researchers find a way to help enhancing that production. And one of the ways of increasing the production is to do a hybrid. This is a hybrid between a channel female and a blue catfish male. This hybrid is going to grow faster. It's going to resist a lot of the diseases. It's going to show that hybrid vigor that aquaculture is looking. You have a better yield at processing plants. Unfortunately, you have to use a blue catfish male. In order for a blue catfish to become mature, it takes around 4 to 6 years. And also, the structure of the testes is not going to allow you to strip the sperm from the fish. The only way to get the sperm is by killing the fish, dissecting the testes, and then smash those testes to release the sperm. Now, Why does cryopreservation wants to do that? Because there is a demand. There is a demand of hybrid catfish of almost 200 millions of hybrid fries last year. So as you can see, there is a demand. And also, you have to know that sometimes if the channel catfish is ready, the females are ready to spawn, probably the male is not ready. So then you lose viable gametes. So by having frozen sperm, you can have a sperm on site at any time. So sperm cryopreservation requires several steps. First, you want to dilute that sperm into an extender solution, which is a saline solution that is going to mimic the seminal plasma. In that way, the sperm is not activated, it's not moving, it's not wasting the energy. You want to remain the sperm inactive. Then you want to do the cryoprotectants and cooling down that sperm cells. After it's cryopreserved, you just thaw it and use it. All these steps require that you have to take a look at the motility, and you want to make sure that that sperm was resisting the cryoprotectant toxicity, was resisting the cooling or cryopreservation and the thawing. Because remember, it's coming up, so it has to pass into also ice crystals whenever you thaw the sperm. And normally, the industry uses these straws. So if you think about it, hybrid catfish, a lot of fish that the industry needs. And we start doing, the normal research by hand, but, there is no way to fulfill this. You have to have an automatic system. So this is my major professor, Dr. Tirsch was my major professor, and Dr. Yang. So Dr. Tirsch decided we have to do it like an industrial scale. So we ended up buying an automatic system that is going to fill the straw, seal it, label, and you can do hundreds of straws in an hour. And also a big freezer to cryopreserve those straws. So okay, now we have the frozen straws. We are ready. So we went to a farm in Arkansas. We induced the ovulation of the females and compare the cryopreserved samples with fresh sperm. And we use the same volumes and pretty much the same concentration of sperm numbers per egg. And we measure fertilization by neurulation. And we didn't see any statistical difference, meaning that you can still use those cryopreserved sperm and you have the same fertilization as a fresh sperm. So that was some success story. Now let's move into the next story. We wanted to apply the high throughput now in another species that is widely cultured in Louisiana. And this is Crassostrea virginica. So in order to develop a protocol, we collected the sperm from the oysters. We dissected and stripped the sperm, the testes, and then diluted in a saline solution. You have to add your cryoprotectant. And we try all these different osmolarities. We try different cryoprotectants, different concentrations, different exposure time. I don't want to bore you about all these details. I just want to summarize what was the results. And we also try different straw systems at different cooling rates. So at the end, we develop a high throughput cryopreservation for Crassostrea virginica. But you may wonder, but you have to kill, that oyster. What about if you can avoid killing the oyster? So that was our next research. Either you can wait until the oyster start spermating and collect the sperm from the water, but the concentration is not the sperm concentration is not that high that you don't have a lot of success doing that. One thing that we were working is any other way that we can collect the sperm from the oysters without killing. Well, we grind a little part in the oyster and do a biopsy from the testes and collect in that way the sperm, and then we just glue it down, and the oysters, the survival varied was 40% survival. It's not that great, but we demonstrate that there is other ways that you can still, without sacrificing the oyster, collect the sperm. Now, but wait a minute. Oysters are Protandry. Are you guys familiar with that? So they start as a males and change the sex after several years into a female. So what about if you can cryopreserve whenever they are little males and wait until they change the sex and self-fertilize that same oyster? So what is the advantage of doing that self-fertilization in oyster? Well, there is a lot of advantage. For an aquaculture setting, you want to have a product that is homogeneous. You want that uniformity. And also it can help you with the genetic mapping. So that's something that Dr. Weeping Yang, the picture that we saw of her, published. So she was able to self-fertilize the oysters. And this is something that, the abalone industry could adopt, something like this. Now, Paniagua, which is in Baja California, in the Cicese, she was able to cryopreserve the trochophores of the oysters. So this is a good news. You don't have just to cryopreserve the sperm of the invertebrates. You can also cryopreserve the trochophores. So the next story that I want to tell you is I was involved in cryopreserving the sperm from corals. So I went to Puerto Rico, in Rincon, and I work with the elkhorn coral. So what happened is coral is endangered. They say 40% of the coral reefs has been lost in the last 40 years. And I work for the Sea Coral Foundation Sexual Coral Reproduction. What is special about this coral is that it just spawn like 1 or 2 nights a year. So the spawning season is like just 1 to 2 days. And the way they spawn at night is beautiful. You can see the bundle, which is eggs surrounded by the sperm, coming from the coral. So we were collecting those bundles. And this was a collaboration with Mary Hagedorn. She works at the Smithsonian. So after you collect the bundles, you want to shake that bundle to separate the sperm from the eggs. Make sure that the sperm has good motility. And start over, mixing with your cryoprotectants. In this case, we were using 10% DMSO or 10% propylene glycol. Because we were in Puerto Rico, we didn't have that expensive equipment to freeze. We have to be adapted to the environment that you are. In this case, you just need a styrofoam cooler, liquid nitrogen. You have your cryo vials ready, submerge them, And in this way, we were attaining 20 to 30 degrees Celsius per minute on cooling. I went for 2 consecutive summers. The second time that I went, what happened is that we arrived and the coral just spawned, like the night previous to that. So it's like, wow, you know, like all this effort on going to Puerto Rico, and now the coral spawned. What we can do? Well, they say, well, there is another colony that you have to go by boat. I think it was like 3 or 4 hours. So we jump into the boat. I jump with the liquid nitrogen, and while they were collecting during night, like in the middle of, I don't know where, so they were bringing the samples, and I was cryopreserving right there in the boat. So, you know, this is just I you know, like I say, I have my dewars with liquid nitrogens, and I was able to cryopreserve all this sperm from the corals. So this means that you don't really need a lab space. Like if you go into a vessel and are going to be spending some time outside, you can also do cryopreservation. Well, the most important part is to demonstrate that that frozen sperm is going to be able to fertilize the eggs. So this is just a copy from the figure from the article that we published. Definitely fresh sperm was better, but we demonstrated that you attain a pretty decent survival on the corals. What we notice is depending on the day that you collect, it's going to depend on your survival after cryopreservation. But we were able to demonstrate that you can use coral sperm, cryopreserve, and produce viable coral. And in fact, some of these coral, I think it's all over the aquariums around United States. So what are the benefits of cryopreservation? Well, you have year-round availability of the gametes. You have a reduction on the needs of maintaining the fish. You are protecting those valuable genetic lineage. And also it can help you with the genetics in improving genetic lines. So what is the future of cryopreservation? Like I say, for fish, everyone asks us, oh, what about the females? You know, what about the females' genome? What about what is going to happen? You are just freezing a sperm. So there is a technique that is pretty recent, and it's called germ cell transplantation in fish, which is collecting the germ cells from a little larvae, and you can use a surrogate species. You don't have to use the same species. And inject those germ cells, which are going to be either spermatogonias or ogonias, into a surrogate species. So now this species is going to develop either the testes or ovaries from the fish that you extracted at the beginning, those are primordial germ cells. So this is something new. I think this is where the cryopreservation is going. And in fact, there is a company here in California that has patents some of these techniques. So that's the toolbox. You know, I'm willing to collaborate in any projects. I want to acknowledge Coast. They pay for my travel to give this seminar. And I want to invite you to come to Humboldt. We have a small fish hatchery, and I started doing some aquaponics too. And that's it. Thank you. Oh, yeah.
You mentioned all the species in the United States
The USDA one? Mm-hmm.
So could you talk about international efforts and how many in Asia and Latin America and such that are being used?
Well, I know different countries like Mexico is trying to have the same germplasm repository. I don't really have the numbers of the species from the other countries. But I know other countries, they start copying the same effort, trying to protect those species. But I cannot give you an estimation on the numbers. So. Yes?
Again, you back to the USDA number. Does that also account or take into account what NOAA has? They've got the big aquaculture center up in Seattle, and the other one over in Milford, Connecticut. I know the Connecticut one is the massive repository of a lot of the oyster staff, the oyster farmers use. So I'm wondering if, like, just because you're focusing on USDA, but may actually be under accounting for, a lot of them might actually be in the repository.
Yeah, this is specifically the percentage that USDA has on the repository. So yes, definitely there is I know Fish and Wildlife also has some repositories on their own, like in Colorado. So this is just an estimation from the one that USDA has. So that doesn't mean that are the only ones out there.
And do they work together? Because I know when I was with NOAA, there's always kind of a source of tension between the aquaculture programs at NOAA and USDA. I mean, has that gotten better, or?
Well, definitely USDA wants something that is farmable, that you can culture. But remember, aquaculture is not just food fish. You can use bait fish, you can use ornamental, so you can justify a lot of the species. Yes?
How does cryopreservation affect the fertilized species later on in life? How do they compare to normal ones?
Yes, and there have been some research about that. And definitely, it's just a normal male, or female, whatever is produced. We were able probably to reproduce like to the second generation, and we never noticed any difference. Mm-hmm. Yes.
So this is mostly to preserve the lines to restock seed stock, right? This isn't, so cryopreservation, it's not as if you're multiplying the sperm, you're simply, so you could potentially do a fertilization that yields a female that could make eggs for you in the future, but you're not going to do this to make a lot of fish from that batch of sperm at that time, right? It's not something where you can multiply the effort.
Well, normally the hatcheries, one of the fertilizations, just take the male and squeeze the sperm into the egg. So there is a waste on the sperm to egg ratio. So there is always excess of the sperm that you can always bank it and use it later.
And is that what it's meaning when it says multiple samples? That's what they are? For example, you had, just on the bison, there's 2 species of bison, but there's 450,000 samples.
Well, because it's talking about all the different lines or pedigrees.
That's what I was after. Is it more about conserving genetic diversity, or is it more about being able to have the potential to create a large production?
Yeah, USA want production, and in fact, if you want the sperm that they have in storage, you can always say, hey, my males are not running, can you send me this many straws? I need to use them, so they can send it back. So it's helping the farmers in just storage and shipping. Mm-hmm. Yes.
Are there any particular fish or invertebrate species that are in really high demand for people wanting to develop cryopreservation techniques for?
Definitely tuna, you know, is one of those. It's very, and it depends, you know, if it's an endangered species or you want an aquaculture species. So you can use cryopreservation as a tool to enhance your genetics in aquaculture, or also to protect endangered species. For example, I was talking about different species that are endangered, especially the small ones. You know, nobody look at those. Delta smell. I don't think there is an effort yet in cryopreserving. So definitely the species are out there, but it's something that I don't know why we don't see the importance of cryopreservation. Yes.
You brought up small things. So can you explain the nematodes? I'm just curious about that. 5% less.
Yeah, I don't really know who is in charge.
It's more than the screw.
More than horses.
I think nematodes are really important in soil health,
Yeah,
I think.
that's what
Yeah.
I was wondering this.
Yes.
Can you speak a little bit more about the potential for the Humboldt State Aquaculture Program in the sense of the species that are of local interest to the Humboldt State Program? What particular species are you working with and might be working with in the future up there, in case those are species that, for example, some students down here might not have access to but might have interest in?
Yeah, historically, Humboldt State University is a salmonids species. So they actually produce a rainbow trout or cutthroat trout. We wanted to start moving into other species, but fish and wildlife is not really open to the idea right now. Another species that they say that we can produce is going to be white sturgeon or green sturgeon. So that's probably something that in the future we want to take advantage. In fact, we were thinking probably the aquaponic system to stock with white sturgeon.
And what about shellfish? Is there any shellfish work going on at Humboldt State?
Humboldt State, unfortunately, is not doing research on aquaculture right now with the oyster industry. But a lot of the students, they go and work for the shellfish industry.
Because the shellfish industry is big in the region, commercially, just not academically.
Yes. Yes.
So let's say one of the species you cryopreserved goes extinct completely in the wild, and all you have are sperm. How would you?
Yes, the same as direct gynogenesis that we saw, you have to use a donor and a species that is closely related, and you irradiate that egg. In that way, there is two options. One, you toll the sperm, you irradiate the egg, and shock them. So in that way, at the time you shock, the mitotic division is not going to release that polar body. It's going to stay and start multiplying. So you can do that way. And recently, what they did is injecting micro-injections of 2 sperms into a donor UV radiated egg. Inject those, so each one is going to form a polar body, and shock them at high temperatures. So in that way, they just came together and you can recover. Then, I'm talking about 3% success. I mean, I'm not talking that you're going to get a fertilization of 100%, but definitely for recovery, 3% is probably good enough.
All right, any more questions? All right, thank you again.